look, here's our studies. So I had to put that into global context. Okay, historical. But here's what we did, and that is we have 5 cohorts of mice. And so one of these we, keep intact. Let's see, is that the intact one? No, not. I know, we overectomized all of these. That is, remove the ovaries. This is 3 weeks. They're about the size of your thumbnail. It's unbelievable microsurgery. Anyway, at 5 months, you put in a new ovary, and these are 2 donor ovaries. So you have to get a mouse that's young, and then take both of her ovaries out and put them into this treatment cohort. And so at 5, 8, and 11 months, we put in new ovaries, and then overectomized control. So one we just, as a control, kept it overectomized, so no new ovary. Another was just intact, so it didn't have any, it wasn't overectomized in a new one. Anyway, there's the surgery. You do the arithmetic on this, is about 500 microsurgeries. Its logistics are unbelievable. And so anyway, this is Shelley Cargill back there. So here's the paper. We made the cover of Aging Cell here. Age of ovary determines remaining life expectancy in old overectomized mice. There's the new ovaries. So this is at 11. This is days beyond 11 months. So we had the what is this, intact right in here, the overectomized. Well, look, we got about a 60% increase in longevity with the new ovaries at 11 months. And so that the 5 and 8 month ovaries didn't do much. And our theory there is simply that young mice don't need to be protected. They're protected by their youth. And therefore, well, we didn't know, where to start. Nobody done this before, so we picked 5 and 11 months. Anyway, we did this again. I'm just going to skip through this. We did it again, and we came up with the same results. This is a follow-up. I thought I was all done with mouse research. Another graduate student comes along, and anyway, we made arrangements to do this whole thing again. While we were doing the first study, this article came out in the New York Times. Tissue transplant, 30-year-old woman entered early menopause, frozen ovaries, then they were reimplanted. She had a menstrual period and so forth. And what I was thinking, they don't know this, but she's going to actually live longer. That was it, and so forth. Now, we did this again. I'm going to skip through this because I've got to keep moving here. But we have 4 more papers, or 3 more papers rather, that came out. And one of these was, let's see, do I have arrows here? Yeah, I have some lines. Look at this one right in here. So that when we did pathologies on this is the second study, is that they found that that is the title here is transplantation of young ovaries restored cardioprotective influence, and so that they're living longer. Well, that's protecting from different diseases and so forth. In this case, cardioprotective. Okay, I'm going to skip that as well because we were going to get into this. These M prizes, like an X prize, they have major money. We thought with this ovary, if we could get funded again, which we didn't, we came pretty close with the Ellison Foundation, but that, this is like 10% of a $3 million pot or something if you increase lifespan by 10% or whatnot. We were going to register our mice and go after this, this is something else. Okay, so there it is, the intervention. So if we made a lot of money, then we did the intervention. Yeah, okay. So there it is. Now look, I know that there's some demography types in here, so I'm going to ask this question. I'll say, life table identity, time to death equals age structure in the stationary population. So look at this. So we have, here we are, out here, say you have even a person, but let's say a fly, individual, you have no idea what the age is. This is out in the field. Now, you capture that, and now you have this captive segment right in here, okay? So you just caught this. You don't know if you caught it one day after it emerged or 100 days after it emerged. You just caught it. But you have this segment right in here, the length of life post-capture. And so that, now question, can mortality patterns observed in the captive segment be used to estimate the life table properties of the wild population? So now you just have this information right in here. What can you do with it? Okay? So we published this paper. It turned out that it actually, you can do a lot. You can reconstruct with the assumption that this is a stationary population, not increasing or decreasing, that you can reconstruct the age structure of that population. So look, here's a hypothetical population right in here, 0, 1, 2, 3, 4, or 3 days. This is, you can't see this, but it's 40%. Is that too high? Well, anyway, it's 40, 30, 25, and 5. Now, let's put a black box. You've just caught all these flies from there, and now they're just going to start dying. You have no idea. They're coming out of this black box. Okay? So each one of these segments you can see up there, they die no matter what age. but they're all dying that first day, so that's what stacks up. Now you do the same thing, these in the box age forward a little bit, I mean 1 day rather, and then they die over that age interval, do the same thing. Then these die, and so forth, and then the last one dies because it's up to the maximal age. Now look at this. You have 40, 30, 25, and 5. 40, 30, 25, and 5. It's a new identity. And I discovered this just as a back of the napkin, just sort of gnawing away at this, when I did some simulations with it. This has to have a mathematical statistical solution, which it did. And so that these are identical, and so that even though that's the original population up there, and this is a death distribution, it reconstructs it. So this is identity. And so that there's my colleague Jim Bopel called it the Carey's identity, lived and life lived, published in Demographic Research. Okay, demographic discovery number 5, call it demographic moneyball. Early MVP, we're talking about the National Basketball Association, is a biomarker for exceptionally long career. I got all the data from the NBA, and they call a life course when they enter the league, and when they, that is, they're born, and when they last contract is not renewed, they die. And so you set up a life course right in here, and what do we have? Yeah, let me go back. And so that this would be this player right in here, which is, and you color code that career each year by the number of baskets he made. It could be assists, it could be rebounds, but in this case it's baskets, okay? Now, you line these up, I call these event history charts, where each career is a horizontal line, the length proportional to the length of the career, and you order them from top to bottom, shortest to longest. And what emerges is a survival curve, right? But also you color code each of the segments according to the number of baskets they made, or points they made rather, that year. And so you've captured the cohort level, survivorship here, but also captured individual level metrics, in this case points. So we have survival here, and the year in the NBA right in here, okay? So look, you can label these as what I call 1 and out, about a third right up here. These are old data. They used to draft just tons of players and let them, Darwinian selection sort themselves out. Now they have a limited number. So this is really dominated by some of the earlier history here. But then you have others, which is 2 to 3 years, promising players cut short, solid players right in here, and what I call an early career sweet spot. This is right in here. Superstars, so you can predict a lot from the first 2 years right in here. Okay? Now look, there's the MVP. So I thought maybe so this is again the survivorship here in the league, and this is 14 years. This is the Michael Jordans, Wilt Chamberlain, and so forth. But you can see right in, and for one, these are the MVPs, okay, throughout history. You'd think that maybe there'd be 1 or 2 up here where they maybe had a career-ending injury, or they got into abusing drugs or whatnot. None. They're all down here. They're all down here with the long lives. So back here, if there's an MVP back here early on, LeBron James, these are, this is somebody to invest in. This is a huge, when you think about it, this is a huge biomarker for, skills and so forth. I mean, to be MVP out of the one out of the entire season. So it's really, really predictive. So that what's happening early in your career right in here is really, really important, especially for MVP. Look, there's all sorts of things you can do with this. I got into this just a bit, as in sort of looking at this. The different ways to look at it based on these event history charts. But I think that there's more than just entertainment here, it's just interest. That really people are aging, from 20 to 30. They lose a step or two from 20 to 25, another step from 25 to 30, and so forth. And this is like Darwinian selection out in the court. Is that this is intense. Everybody wants to be out there and playing, so that not like, if they're slackers at all, they're out of there for the game and maybe out of there for the season and career. So that I think that even studying data like this, it'd be has potential for informing aging research. Okay, I'm going to keep burning through this. So anyway, that's early in this. What is this? Peak. This is a percentage of top players points. And 47% is the peak, but then they fall back off here. They start out right in here, so that rookie years and so forth, they start out by the end of the year, they're back at the rookie year level. But then they're back at their off the team. Okay, I'm going to skip through this. Number 6, current frailty and future longevity depend on the past nutritional conditions. I just slipped this in last night. And so anyway, what we did, this is a big part of aging, as Peter would know, and that is, what does early life conditions, how does this play out when you're 40, 50, 60 years old? And in fact, even before that, it would be the fetal origin hypothesis, this Barker hypothesis, where in the womb, if there's nutritional issues, that is shortage and so forth, is that when you're 40 and 50 years old, it's what your mother's nutrition that played up higher blood pressure, higher heart disease rates, and these sorts of things. But anyway, so what we asked was, what's if we, the nutritional conditions early in a fly's life, or for the first 55 days, how would that translate? This gets a little more complicated. I might sort through this. There it is. Burn through it. Let me see if I can just get to the summary here. Okay, there it is. We have in two contexts here. Two results that I'll talk about. One would be we had group cages there. You saw those big group cages, and then we take out at 55 days 1,000 flies and put them in individual cages. Okay, and they were on either sugar diet or full diet. But getting them from the cage to the big cage to the small one, there's a transfer stress. Okay, so they use an aspiration tube and transfer. And so all of them were subject to that same kind of stress. But you look at this, and you have look at this, the past diet. So if they came out of sugar only, this is frail. 40% of those died within just the first few days because of that transfer stress. If they were on a full diet, it was only 20%. And so the previous diet really made a huge difference. And then furthermore, here's we get back to males and females anyway, is that I guess I have summaries over here. That is 20% or 21% of the females overall died from that transfer, whereas almost twice as much for the males. So the frailty overall, male-female. I'm going to skip this and get to a couple others. Okay. Here's one. Longevity extension is self-reinforcing. The concept here, and this is connecting entomology and insect biology and so forth with humans, is where I'm headed here. And that is the evolution of sociality in wasps. Here's the model, and that is that you start out as solitary wasps. It's not quite solitary. This is a couple of stages out. But anyway, individual wasps, they're basically laying an egg on like a caterpillar in a hole right in here. There's no sociality. Then you have the emergence of parental care right in here where they're feeding their young. And then right in here someplace, this sociality threshold where the mothers are alive at the same time as their offspring, that is the daughters. So now you have incipient sociality, so that now you have division of labor and so forth. So the key here, the carry away concept, is that that longevity was a precondition for the emergence of sociality. But then you have, with greater longevity, you have greater sociality emerging from that, so that they're self-reinforcing. So that the greater longevity, you have more division of labor, these create nests and so forth that can protect them more, reduce mortality even further. Anyway, it feeds, it's a self-reinforcing system. Okay. Now the main point, extended longevity is a precondition for evolution and incipient sociality. Sociality in turn creates conditions for the evolution of further extensions in longevity. Okay, these are self-reinforcing. Okay, general theory of longevity. We took this to humans, where you have increased child survival. It reduces the number of children sought. We know this. So that instead of having 12 kids, you have 6 kids to run the farm or whatever. This turns in, because you have a reduced number, not gestating, pregnancy of 10 or 12 pregnancies, at least for a wife. You have healthier, long-lived parents, grandparents. Okay, this translates to better educated. So I always say that instead of having 14 high school graduates, you have one Iowa State graduate in molecular biology or something, and that you can't get the same place with 14 high school graduates that you can with one molecular biologist or whatever in terms of advancing technology, for example. So you're putting all your resources into a smaller number of offspring. This feeds back on itself. Anyway, you get the idea. Okay? I'm going to skip that too. Sorry.